Thank you very much, and thanks to the organizers and the work package leaders for the invitation. It's a great pleasure. And I just realized this looks like a smiley. Well, yeah, I will talk about organomics to study human development and disease. So we are excited by human development and the large diversity of cell types that is being generated during this process. And accordingly, we are very excited about the new advances in the field of stem cell biology, which allows us now to take pretty much any a somatic cell of any human individual, generate these induced pluripotent stem cells from them, and then differentiate those not only in two-dimensional culture into various lineages of the human body, but also in three-dimensional culture into these organoids. And you've seen a lot about that already, especially Jürgen gave a great intro for the slides that will be coming. What I want to mention as well is we can actually not only reprogram a somatic cell into a pluripotent stem cell, we can also directly reprogram a somatic cell into another somatic cell, such as a conversion from fibroblast to neuron, which can be quite handy as well to model disease, because it was shown that kind of age marks in the cells in fibroblasts of aged human individual are being maintained during this direct reprogramming, and they are lost during this going through the pluripotent stem cells. And so we are excited by these organoid systems, and of course they have this promise that you can model an individual human's disease in the dish. And a lot has been done in that regard already. There are quite some papers and studies out there where various diseases were modeled using organoids in various organ systems, and this is a review from the Cleaver lab. I should point out that there are different types of stem cells, ASCs and ESCs, or iPSCs, and the ASCs are the adult stem cells from which you can make organoids, and this is what we saw in Priska's talk, in Alexander's talk earlier today. And so these are from adult stem cells that you derive organoids, whereas we are focusing a lot on these iPS or ESC-derived organoids that really start from pluripotent cells, and in this case, model disease as you grow the tissue. Whereas, as Priska showed, that if you start from adult stem cell, you might rather go through a regenerative procedure to make a tissue and then really mimic the maintenance of an adult organ. And but I want to take a step back from this and take some thoughts, or that's what I did when I prepared the slides, on what we need to consider when trying to model disease in organoids. And one thing that we really need to make sure is that our organoid system accurately and precisely recapitulates the cell states that we want to model the disease in. So here, if you consider this kind of a fate space, it could be the organoid or another in vitro system is not really accurate or precise. It generates all kinds of cell fates that are not recapitulating what you want to do. You might be accurate such that on average you have the right fate, but it might not be precise because you have quite some variability. On the other side, it could be precise but not accurate. And of course, this is what we want. We want an accurately and precisely modeled cell state, and all the states that are in the organoid should be like that. We need to consider variability across organoids, organoid batches, lines, and human individuals, since these are confounding factors, such that if there's too much variability, we might just not see a disease phenotype. And then we also need to consider or make decisions in our experiments whether we want to really model the disease of an individual patient, and meaning we take that patient's cells, make induced pluripotent stem cells, and then make organoids, or whether we actually take our control line that we know works very well in our lab, and we use CRISPR-Cas9 to precisely edit that line to introduce a disease mutation. In that case, of course, we have an identical genomic background of the control line and the mutated line, but we don't really have exactly the genomic background of the patient. And then we should consider which in vitro system suits which disease. So if we want to study some late onset disease, it might not be good to start with pluripotent cells and early in development, and the other way around. And then, of course, we should consider whether a disorder can actually be treated when we then move from the organoid system to the patient, and what diseases are maybe more amenable to treatment or to development of therapies. And so what we think is that single-cell genomics have really a set of powerful tools to make this assessment whether the organoid systems or the in vitro cell types and tissues that we generate are precise and accurate. And of course, as an effort in the Human Cell Atlas, we are generating a lot of atlases from primary tissues during development, adulthood, aging, and we can compare these, the single cells from these atlases, to single cells that we measure derived from the organoids or some other 2D in vitro system. And we can do this on the transcriptome level, which is of course the most easy and feasible, but we can of course also do that on a spatial level and using epigenomics and other methods. And if we now focus on transcriptomics, what could come out of such a comparison is that, for example, here you have different clusters you detect. Your primary reference is in black, the organoid in light gray. You might see that a lot of clusters nicely match, but there might be lineages that are missing in the organoid, but they're in the primary tissue, and then you might have off-target cells in the organoid that you really don't see in your primary tissue. And so this is then what you can find out, and this allows you to maybe go back and reverse engineer and better engineer your organoid system. What you can do as well, what we can do with single-cell transcriptomics is reconstruct differentiation trajectories, and in this case we can map a line in vivo trajectory with an in vitro trajectory and find out how the timeline matches and analyze similarity of cells. In this case, for example, in a pseudotemporal expression profile of certain genes, we might find out that in the in vitro tissue, gene 4 is not expressed in a way, doesn't come up at later time points as it does in vivo, and this might show us how our in vitro system doesn't mature as we would expect from in vivo. And finally, of course, single-cell genomics is not limited to these simple technologies such as transcriptomics, ATAC-seq, and so on, but there are new tools that are developing, and we have heard about this today already, such as from Alexander, we've heard about lineage-coupled single-cell transcriptomics, and this is, of course, what we can do in organoids, especially because we can manipulate them. We can get into spatial information by doing niche-seq or spatial transcriptomics, or infer it from transcriptomes by receptor-ligand pairing analysis. We can do CRISPR-Cas9 screens in high throughput such that we don't need to generate isogenic lines in a tedious way, but we can generate mosaic organoids. And because of a single-cell analysis, we can then dissect what the effect of a certain gene knockout is on each cell in the organoid. And this is a bit more general. We can also multiplex transcription factors that can be overexpressed with the attempt to engineer a very precisely a cell fate that we couldn't make in vitro so far. And in this case, the single-cell readout will also allow this to be done in high throughput. And so I will show you a little bit what we have been doing along these lines. We have focused quite a bit on cerebral organoids, and this will be most of the coming slides, also on liver organoids. And for the cerebral organoids, we really started using them in the lab. We use Jürgen's, Madeleine's protocol, and we were curious how similar are they to a primary developing human cortex. And so we dissected out fetal neocortices and organoid cortices, really focusing on these nice cortical regions that Jürgen also showed earlier. And then we performed single-cell transcriptomics, in this case still lower throughput methods, in this case C1. And when we put all cells together in this intercellular correlation network, we could see that the cells from independent of the tissue of origin very nicely mixed, which showed us that the organoid cells and the fetal cortical cells are really highly similar. Also looking at the set of genes and their pseudotemporal expression profile showed us that the cells really nicely recapitulate the gene expression profiles that are found in vivo. And in our liver work, this liver organoid protocol that really also mimics liver development is based on generating hepatic cells from iPS cells in vitro. And then these hepatic cells are being co-cultures with endothelial and mesenchymal cells to generate these organoids. And what we did is we compared the two-dimensional differentiation of these hepatic cells to this differentiation within the organoid. And when we put all cells together here, what we saw was that in this lineage trajectory analysis, we saw that initially the cells are very similar in 2D and 3D, but as soon as you put cells, let cells differentiate further in the three-dimensional tissue, they become quite different and mature on a different trajectory than the cells in two-dimensional monoculture. And what we could show is that actually the cells that are growing in the organoid and that have different lineages around, such as the endothelial cells and the mesenchyme, they are more similar to a primary developing liver than these cells in 2D culture. And this really highlights that it makes sense and it's worth studying developmental biology in organoids as compared to a 2D differentiation. And so what I showed so far was really a bit limited in throughput. We captured only 100 cells per experiment. And what we wanted to do next is really in high throughput analyze variability in these organoids and the cell types that are emerging. And not just cutting out such a cortical region as we did before, but really looking at the whole thing. And we started by really looking across the whole development from pluripotent cells to 4-month-old organoids. This just shows how, if presented by time point, how the heterogeneity increases over time, starting from one cluster to various clusters at 4 months. And this is taking all the data together and doing such a KNN graph from the data, where we then see how from the pluripotency the cells differentiate through neuroectoderm and neuroepithelium, and then diverge out into various different trajectories, lineages, one might want to say, but it's of course not a direct lineage measurement. And what we could show is that, here looking at FOXG1, that this and this branch is really the telencephalon, and we can distinguish between the cortical area, so the dorsal telencephalon and the ventral telencephalon, the GE-derived neurons. We have also hindbrain cells in there and some midbrain neurons. We have astrocytes that emerge at 4 months. And then we have always a few mesenchymal cells, and in this case also some retina developed. And we can also here calculate the similarity to a primary reference, where we very nicely see these cells are really the cortical cells. These are the GE-derived cells, such as the early striatum, and here are cerebellar cells. And we can also map the individual cells from the organoid to a primary 3D map. I mean, here it's shown just 2D. And then this helps us to assign what cell type actually we have here and what the regional identity might be of each individual cell. And you can see that we capture cells really across various regions of the brain. And so I talked about variability in the organoids, and what we did here is we generated cerebral organoids from 7 different lines using the HIPSI resource. And when we put here all the data together, again, we see 3 major branches starting from neuroprogenitor cells into these neuronal lineages. And they are annotated here with, again, ventral forebrain, dorsal forebrain, and then here all kinds of midbrain and hindbrain neuronal populations. And here's how the 7 different lines are distributed across this plot, which shows that overall, I would say, all the different branches have some cells of each line, but you can also appreciate there's quite a variability, such that, for example, KUCG in this case mainly makes cortical cells, and there are other lines that very nicely make all the different cell types. And so this summarizes this variability, where if you focus just on WIBJ, I can actually tell you that these 2 organoids, the first 2 are 1 organoid batch, and the second 3 are another organoid batch. And you see how in the first batch, this line made mainly ventral forebrain cells, also a little bit of the other regions, and then in the second batch, mainly cortical cells. And so this is, of course, a variability that we really need to consider and try to improve, and Jürgen talked about that a bit, and he also saw that based on stainings, actually. But what I want to highlight, and this is what we are excited about, is that we now, if we now look at pseudotemporal expression of certain marker genes, such as GLI3 for the radial glia, EOMIS for intermediate progenitors, NeuroD6 for these early-born cortical neurons, and GRIA2 for a bit more mature neurons, you can see that across the lines, this expression is quite reproducible, actually. So once we identify with the single-cell analysis the identity of a cell and the regional identity of a cell, then really across the different lines and individuals, this is quite reproducible. And so we are excited about this system to model disease, and we started looking at malformations of cortex development, which are generally due to abnormalities in cell proliferation and neuronal migration. And there are different patient phenotypes, including intellectual disability, epilepsy, and autism. And some causal genes are known, but oftentimes mechanisms are really not understood. And we chose one malformation that is periventricular heterotopia, in which case there is a subset of neuronal cells that fail to migrate to the cortical plate, so they are stuck at the ventricle. And so we generated iPSCs from control cells and patients' cells. And then we were very excited, and this is in collaboration with Silvia Capello at the MPI for Psychiatry, that actually we see in the mutant or in the patient-derived organoids these nodules of neurons clustered at the ventricle. These neurons failed to migrate, whereas other neurons actually migrated in the right way. And in control organoids, we don't see this. And then we did single-cell transcriptomics, where we then found that generally we have a differentiation from progenitors to neurons through intermediate progenitors, but only in the mutant organoids we find a subpopulation of neurons on this altered neuronal state, which branches away from the normal neurons. And this is really what we then want to focus on to look further into this disease and maybe even find a way to manipulate the development. And another disease is GCPS, which is another disorder that induces brain malformations, in particular macrocephaly and phenotypes as, for example, mental retardation. And it is due to a loss of function mutation in GLI3, which is a gene that really is expressed in the progenitors. And what we did here is the other way to mutate, to model disease by not taking a patient cell and reprogramming it, but taking our control line and editing it to introduce the mutation. And then we grew organoids for both the control organoids and the GLI3 mutant with the same genomic background. And this is what we get. You can see in colors control and mutant, and again, annotation with mid and hindbrain, ventral forebrain, and dorsal forebrain. And it was really striking that we don't see any mutant cells on the dorsal lineage, whereas they are very nicely represented on the other lineages. And so this really shows that GLI3 is required to generate the cortical neurons and progenitors. And this is consistent, for those that know more about that, this is very consistent with previous work in the mouse, where knockout of GLI3 really reduced the size of the cortex and made ventral forebrain cells migrate into the dorsal area. And so generally, we are very excited about these organoid systems. We showed that they can recapitulate genetic programs of the primary organ development, and this is, of course, different to the adult stem cell-derived organoids. We showed that transcriptional trajectories are reproducible across human individuals. We can model malformations of brain development, and we can actually analyze mechanisms of fate determination, which was shown with this GLI3 knockout. And here I'm kind of repeating what Jürgen said, and I'm out of time, so I should go very fast. But there are shortcomings to the organoids. I don't want to not talk about that. Which is that there are missing lineages, and it was pointed out. Microglia, for example, the vasculature is missing. And these are 3 different organoids, and you can see really each organoid is different from the other. And we would eventually want to have a very stereotypic organoid development to be able to directly see a disease phenotype, more or less, in the case of the malformation, and not have to go through hundreds of organoids and then apply statistics to find out whether there is some difference. Because in the primary human individual, in an MRI scan, you directly see the malformation. And so that's why I think within the lifetime consortium, we should also kind of think about really enhancing organoid technologies for disease modeling, which would mean, for example, controlling the patterning of organoids and of the morphogenesis for having a stereotypic development, analyzing the line-to-line variability in way more high throughput than we did so far, and then really have interlineage crosstalk in the organoid, which is important not just for development, also for cancer, to not just have epithelial cells, but also the cancer niche with the mesenchyme and the endothelial cells, the vasculature. And have the organoids, and this was a great talk just before this talk, to really be able to perfuse organoids and not just have a static vasculature, but actually use it for perfusion in microfluidic chips, for example. And so then I think it would be super exciting to have multi-omic single-cell analysis of individual humans' disease with the goal to develop therapies. Thank you.
Thank you, Barbara. One quick question here.
So you showed these very nice 4-month-old organoids, and I was wondering what kind of connectivity do you get in these neurons, and how far does it reach? You said it was like fetal-like stages, but what kind of stage?
Yeah, I must say that we did not assess the connectivity in this case. We really focused on a genomic analysis or transcriptomic analysis of the tissues, but others have shown that there is definitely connectivity that, like in Paula Alotta's Nature paper, for example, Quadrato et al., they showed, for example, that this is being set up in these organoids.
One last question here.
I would like to know if you reach the myelination states in your organoids, and do you have ependymal cells on the ventricle or around the ventricles?
So the first question was myelination?
Yeah, do you reach the myelination states, and do you have ependymal cells also?
Yeah, so we, for example, don't see oligodendrocytes in our analysis, and of course, by now we can capture many thousands of cells per organoid. It could be that the media composition that we are using, and this is definitely something that needs to also be discussed, favors really the development of neurons and astrocytes, but not other macroglial lineages. As far as I remember, Jürgen sees sometimes in stainings oligodendrocytes develop in the organoids. Yes, you can see that. Yeah. When you wait longer, then also oligodendrocytes should come up, but at 4 months we don't have that yet. We have a small proportion of astrocytes. We also do this for other species such as chimpanzee and macaque, and there actually we see these other lineages coming up much earlier because there the development is faster. So, but for the human ones we need to wait much longer.
And about the ependymal cells?
We have never seen them yet either. Yeah, I think these are all our missing lineages where we need to assess whether the time point is, at that time point that we analyze the organoids, they would be already expected, and if so, then we really need to put effort to have them co-develop or have them infiltrate the organoid artificially.